So we're back on our evidence for evolution. We are on number 5. And at the time they were tripping over fossils, they were also dissecting organisms, many for the first time. And they started to see variations on the same theme, as Neil Shubin likes to call it. They're starting to see long bone, 2 skinnier long bones, but to bones, little bits. And they're seeing this over and over and over again. And Darwin realized that it wasn't, nature just doesn't reinvent structures. It takes a structure and varies it. Okay, and the forelimb of all vertebrates is a great example of that. And these are known as homologous traits. Homologous structures. This is not in your student PowerPoint, but I think it's important to point out that you cannot always think that they're homologous. For example, shark dorsal fins and dolphin dorsal fins are not descending from a common ancestor, but rather because they are in a similar environment, they are developing similar features that help them survive. And these are called analogous structures or convergent evolution. Embryonic development is also strikingly similar among organisms. It's almost like we go through our stages of evolution as we go through our embryonic development. And this is a funny thing. There is a point in human embryonic development that we have gill arches, which become the gills in fish. And some of us have a congenital malformation where we have a gill. And I have a gill right here. I'll show you at lunch. So lunch is 3 special things. I always wondered what this was. And it's gotten infected. I got infected once when I was a little girl. And it's basically, you all had this at one point in your embryonic development, but mine never closed. Now, I wish it worked. I wish I could actually breathe underwater because that would be amazingly cool, but it doesn't. It doesn't actually work. But I have a gill here. It's called a preauricular fossa is the scientific term for gill. My gill, anyway. Vestigial structures. Scientists began to see that organisms had structures that have no use, but yet they had use in the ancestor, and these are known as vestigial structures. I've had ball pythons in my classroom. They have anal spurs near their cloaca, which were legs. The whale, you cannot see it because it's covered by blubber and skin and so forth, have hind legs, and we have goosebumps. You know, why would we have goosebumps if we didn't have fur? Must mean that our ancestors had fur. So those are known as vestigial structures. Yes?
I don't know if you've all seen, but I just got some what are called mud puppies. They are the salamanders,
Right.
that have both gills and lungs.
Axolotl?
No, not axolotls, which change. These don't. These are from Michigan and North Carolina. And they can never really come out of the water, but they actually have the whole set of gills and the lungs simultaneously. As they develop, they retain both. They have immature lungs.
Wow.
And immature gills, but they need both to survive. They're called, I don't know the scientific name, but they're called, but they are native to the United States. They're cold water from North Carolina to Michigan in the waters. And
Your classroom, I'm sure, is a very fascinating place.
We have about 15 inches. We have 2 young ones that are about 10, and the adult is 15.
Wow, very neat. I want to stop and do a video here. We don't just see evolutionary history out there in the world. We carry it within us. We carry evolution within our own bodies. Have you heard of the fascinating case of the recurrent let me try that again. The fascinating case of the recurrent laryngeal nerve. For those of you who are a little queasy, it is a giraffe dissection. It's only been done once, over 150 years ago, and then this time. And what you're going to see, as you can see from the picture, the nerves that innervate our voice box. On one side, it goes from your brain to your voice box. On the other side, it goes from your brain, down your neck, around your aorta, and back up to your larynx. In a human, that's kind of weird, but in a giraffe, it's ridiculous. So we are going to see an amazing anatomist dissect a giraffe, and I'll let Professor Richard Dawkins take over here.
We get used to the idea that evolution is so good at producing beautiful, elegant animals that look as though they've been designed. We forget that sometimes they're not perfect and there are imperfections. And the imperfections are very revealing because they're exactly the kind of imperfections you'd expect from the accidents of history if there were no designer. There's a nerve called the recurrent laryngeal, which runs from the brain, and its end organ is the larynx, and you think it would just go straight there. But in a human, what it does is goes down into the chest, loops around one of the main arteries in the chest, and then goes straight back up again. Obviously a ridiculous detour. No engineer would ever make a mistake like that.
In our bodies, the laryngeal nerve takes a circuitous route, but what happens in an animal with a neck as long as this? It's one of the great evolutionary enigmas that Richard Dawkins is keen to resolve in The Flesh.
Interestingly, it's never been dissected out, or only once before has it ever been dissected out in its full length, and that was in 1838.
Don't cut it at this crucial moment.
I'm too good an anatomist for that. You have to trust me now. What I'm actually holding here is the beginning of that nerve. It actually starts out not as a separate nerve, but as a branch coming off of a bigger nerve called the vagus nerve. And this is going to keep running all the way down the body. So you'll see it again over here, all the way down the neck on both sides. So you can see it again right there.
Yes.
And this is going to wrap around the great vessels coming out of the heart. So here it is wrapping around that, and then it continues right there. So here's the vagus going down, and here's the vagus continuing.
Right.
And right over here, there's a branch right there. So it's looping and it's coming back, it's doing a U-turn all the way down here. So it's traveled that entire distance to make a U-turn to go all the way back up again. And so now we can follow it going back up again. So we follow this branch, and if we look, we see it again over here. There it is, like that. And here you see it going up. This is the voice box, the larynx, and so you can see it going right into the back of the larynx here. And as it innervates this area, it controls the muscles that then control making sounds, but also coordinating breathing and swallowing in this area. So this is a very important nerve. Interestingly, where it ends is pretty close to where it started. It started here, coming out of the brain. It really only needed to go about 2 inches.
Yes, amazing.
But it went all the way down and came all the way back.
Yes. That's a beautiful example of historical legacy as opposed to design.
Exactly. This is not an intelligent design. An intelligent design would be to go from here to here.
Yes.
It does kind of beg the question, though, that even in an animal that might have been many millions of years ago with its head down here, why the route round the blood vessels, unless there was a reason that they were there to innovate something else?
Well, that was in earlier ancestors. Then it was the most direct route in fish.
So it's just inherited.
It's a historical legacy.
Fish really don't have much of a neck, so there isn't any extension to worry about in fish. It just goes directly there. Once you introduce a mammalian neck, you start to get a longer neck, now the heart is displaced down lower.
It turns out the laryngeal nerve first evolved in fish-like creatures as a direct link from the brain to gills near the heart. Over millions of generations, this nerve gradually lengthened, each small step always simpler than a major rewiring to a more direct route.
Remember that a designer, an engineer, can go back to the drawing board, throw away the old design, start afresh with what looks more sensible. A designer has foresight. Evolution can't go back to the drawing board. Evolution has no foresight.
Could I add something to that?
Sure.
Because they mentioned the vagus nerve, Texas A&M University. One idea or thinking of that is that it is related to the heart. If you can actually slow your heart rate down by touching your eyeball, because your eyeball stimulates the vagus nerve, slows the heart down.
Right, but that's the recurrent laryngeal nerve goes down with the vagus nerve, but the other one doesn't. The other one just goes straight there. Interesting. I don't know how is that appropriate for 7th grade with the giraffe? You might get a few.
I think so. They can make those sections. Yeah.
Okay. You know, I did show it. My 8th graders are taking 10th grade bio. There was a little pause because, especially at the end there, you know, you got to know your children if you want to show that video.
It's been good technique.
Mm-hmm.
It's different when that zoo butchered that giraffe to feet
Oh,
to the
yes,
lion, that's a
of
little
course.
different, that would be. But they seemed very respectful, it was a
Yeah.
good technique, good way of doing
I'm
it.
going to assume the giraffe died of natural causes. I don't know.
Does any other animal have the,
All vertebrates have that circuitous root. It's just in a giraffe, it's so obvious. But we
so
have
that's
it,
why they
dogs,
use the
cats,
giraffe.
everything. You know, it's the same thing with the vas deferens, the testes, vas deferens, penis. One of them goes around the ureter. One of them goes directly to testes, penis. The other one goes up around the ureter and back down again because testes were in the abdomen of fish. So same idea, but I'd rather do this with the kids because you guys are teachers, I can tell. I'm just going to stick with the name. And finally, my favorite part of teaching evolution to my students is molecular biology. This is really changing the face of evolutionary studies. This is the strongest evidence for evolution. Darwin did not have this information. As a matter of fact, Darwin did not read Mendel. Mendel read Darwin, but Darwin didn't read Mendel. And he had some misunderstandings in terms of how traits are passed on in organisms. He believed in kind of like a blended inheritance, and we understand now that that is false. And these, when I talked about homologous structures, when I talked about the arm being a homologous structure, it's the same in humans as it is in a bat, as it is in a cow. Genes are also homologous structures. And now that we understand the genetic code, we can compare the genomes of different organisms and see how amazingly similar some of them are. So I don't know, I think you teach evolution before genetics in 7th grade. So I threw in some slides for you just to give them an introduction for genetics. Something like, so what is a gene? This is from your inner fish, by the way. I showed them the structure of DNA. So you could give them a basic understanding for later about how, this nucleotide thymine always matches up with adenine. Cytosine always matches up with guanine. I tell my students, act Coral Gables. Or act Coconut Grove. And by the way, when I get to RNA, since I have a lot of French kids, I say, oh, Coral Gables, because it's uracil replacing thymine. But anyway, that's high school bio. So basically the genetic code, the students need to understand that DNA has 4 nucleotide bases, and that the same one always complements another. It's called complementary base pairing. A T will always be with an A. So there's your T, there's your A, there's your A, there's your T. A C will always be with a G. A G will always be with a C. And we have now sequenced not just the human genome, but many, many other genomes. And we can compare. And if the human has a GC, at that specific point in the chromosome, in the DNA, what does a chimpanzee have? What does a mud puppy have? What does our silver fox have? And what we're finding is we have tremendous similarity across species. Even in something like a fly and a human, there are genes called Hox genes. They organize you as you are an embryo. They put your head this way and your feet that way and your arms that way and everything else. And we have found Hox genes that correspond to human Hox genes in the fly. They've done some amazing studies. For example, they have taken in a growing embryo of a chicken, they've taken the area that's supposed to be the forelimb, which will become a wing in a chicken. And they take that tissue, it's called the zone of polarizing activity, and they take that tissue and they put it in a growing embryo of a mouse at that exact same point of embryonic development. Does that mouse get a wing? No, that mouse gets a perfectly formed mouse forelimb. In other words, the mouse cellular machinery can read chicken DNA and make its forelimb, because that's how similar the DNA is. And they've done this with different organisms. And this, if you're interested in this, it's in your inner fish, the book Your Inner Fish. I have here a couple of very interesting videos. We'll do these now. The first one is Richard Dawkins comparing the human DNA to chimpanzee DNA. And the second one is the author of my textbook, Kenneth Miller. He wrote the textbook that we use here in Miami-Dade County for biology in high school, the one with the parrot. Do you have that book? And he's going to show you something really cool. We have one less chromosome than all the other apes. if you understand genetics, that should be lethal. That should be absolutely deadly. We could not have, at some point in our evolution, lost an entire chromosome. As it turns out, we did not lose an entire chromosome. 2 chromosomes fused together. And that is our human chromosome number 2, corresponds to 2 smaller chromosomes in the other apes. And we have actually found exactly where that fusion point takes place, down to the nucleotide base pair, and I'm going to show you those 2. So we'll start with the less complicated one here. And again, all of these resources, I'm giving you the links in the back of the PowerPoint. It's this one. There's other ones here, but I like this one the most.
Humans and chimpanzees parted company in evolution about 6 million years ago from a common ancestor. The human genome and the chimpanzee genome have both been completely sequenced, and this is a small section of part of the genome that matches. If we follow along here, human chimp, go all the way along here, all the way along here, they're all identical. Differences between the two are indicated by a little man who is in fact Svante Parbo, a very distinguished Swedish geneticist now working in Germany. It's a little game of find Parbo. Follow along this row here, chimp, human, identical, identical, identical. There's a difference. Walk along, walk along, walk along, all the same, identical, no difference at all between the human and the chimpanzee, exactly the same, exactly the same. Now we see another difference there, A versus G. Walk along, walk along, walk along, and there are no more differences in that whole row. Almost all of the human genome and the chimpanzee genome is identical. A tiny number of differences account for all the really quite large differences that we see between humans and chimpanzees.
He's going to explain how our chromosome number 2 is actually 2 chromosomes fused together at some point.
When 20th century scientists discovered the role DNA plays in heredity, they founded a new science called genetics that put Darwin's theory to the test. The cells of all great apes, like chimpanzees, gorillas, and orangutans, contain 24 pairs of chromosomes. If humans share a common ancestor with apes, you'd expect us to have the same number. But surprisingly, human cells contain only 23 pairs.
Now, if we share a common ancestor, what happened? Is it possible that in the line of evolution that led to us, a pair of chromosomes from a primate ancestor just got lost and just got discarded? Well, the answer to that is no, it's not possible. And the reason is because every primate chromosome has so many important genes on it that the loss of both members of a pair would be fatal. You wouldn't even get through embryonic development. So there's only one possible explanation. And that explanation is that in the line that led to us, 2 chromosomes that were separate in other primates became fused to form a single chromosome. And this is why evolution is a science. That possibility is testable. We ought to be able to look at our genomes and find that we carry a chromosome with the marks of that fusion on it. Now, how would we find that? It's easier than you might think.
Typically, on the ends of every chromosome, you should find special genetic markers, or sequences of DNA, called telomeres. And in their middles, you should find different genetic markers called centromeres. But if a mutation occurred in the past, causing 2 pairs of chromosomes to fuse, we should find evidence in those genetic markers. Telomeres not only at the ends of the new chromosome, but also at their middles, and not 1, but 2 centromeres. Finding a structure like this in our chromosomes would explain why humans have 1 pair fewer than the great apes.
If we don't find it, then the case for common ancestry for our species might be refuted. But if we do find it, it would be powerful evidence in favor of evolution. So all we have to do is to scan the human genome and see, do we have a chromosome that has these marks, telomere DNA in the center and 2 centromeres? Well, the answer turns out to be we do. And it's human chromosome number 2. And the evidence is unmistakable. We've suspected this for a very long time, and in 2005, a definitive study was published showing that chromosome 2 has the exact fusion point, almost the point at which the Scotch tape holds those 2 chromosomes together can be found. The closer we look at our own DNA, the more detailed a glimpse we get of our own genome, the more powerful the evidence for our own evolution from common ancestry with other species becomes.